Okay, so let me start today's week's discussion, and if someone joins, I will let them in. And since it's already 6 p.m., so welcome to the week 5 structural biology course of NPTEL. And so today, what we will be doing is that we will be discussing about the week 5 contents, which has been already shared to you by the NPTEL. And also, I will be solving the week 3 assignment. Okay, so this is the general overview of today's class. Now, what we will be doing is that if you guys have any doubt or questions, you can directly write to me in my Gmail ID, or you can put your questions in the discussion forum, or you can actually ask me in the next class about any doubts about the assignments that you have. Okay, so since so what I can discuss and start this session with is that I can discuss some questions from the last class. So remember we were actually talking about the so I think now the slides are in full for here full full view. So remember from the last class when we were talking about the different kinds of symmetry operators that is very useful and handy while actually solving the structure, because in a crystal there are different unit cells, and inside the unit cells you have this your lattice planes which consist of different atoms. Now if it is very easy to decipher the positions of all the atoms in the crystal if you find any pattern associated with that with one molecule to the other. Okay, so for that what you can do is actually that you can actually use the some kind of symmetry operators like reflection, glide plane, or you can also you have also seen about the rotation operators. So we have talked about this, but what we were discussing is that six-fold symmetry. Six-fold means if you divide the 360-degree angle divided by 6, that is 60 degree, that each point is associated to another point by a 60-degree angle. Then it is means if you draw 60-degree angles and you actually complete 360 degree, you will actually complete all the atoms which are associated with that single atom. So then that atom is connected with other atoms with six-fold symmetry. So what we discussed is that among all the folds of symmetry, the six-fold symmetry is more stable as compared with others. So why that is so? So the question this was also asked in previous year. So I was just wondering why that is so, and I have looked into it. So the main reason behind that is that if you see the laser pointer here, so if you see this is the shape of your pentagonal shape, okay, and this is your six-fold, okay. So we have, for example, why six-fold symmetry is more favorable. So why therefore looking into that, I have taken the example of a hexagon and a pentagon. Okay, or which is similar to an analogy. If you take, this is similar to the five-fold symmetry, and this is similar to the six-fold symmetry. So when you are actually for X-ray crystallography, what is important is that we require a crystal which should be in which the atoms should be ordered in a order or perfectly arranged in a order. So to maintain that order, it should have a high symmetry, and also there should be minimal empty spaces inside the crystal. That by that I mean that the crystal should be properly packed. Then only it will be a properly ordered crystal with periodic unit cells, means periodic boundaries satisfying it, periodic condition satisfied it, that is there should be a periodicity between each unit cell in the crystal, and that should be only maintained if the space inside the crystal is minimized and the packing is efficient. Now if you see that if two crystals are made, one with this pentagon and another with one with this hexagon, in this hexagonal unit cells you see the spaces between the hexagon is very minimal. Whereas in this pentagonal shape, you see if you add more pentagons in it, then the spaces between these unit cells or spaces between these unit cells which will be forming the crystals, they there will be very large spaces, and this will actually lead to various irregularity, and it will hamper the orderness of a crystal, and this will be actually hampering whether your proteins will form the crystal or not. That's why the six-fold symmetry is mostly favored in your crystal, protein crystal. Okay. Another question is why there is a lead coating around the X-ray diffractor. So for that, so Shani, right? Okay. So I have just started the class, and I was discussing. Uh, yeah, Shani. So if you have any doubt from the previous session or any assignments from of previous assignments, you can ask me. Okay, then we can start today's discussion. Okay, okay, so that's fine. So you will find the recordings of all those sessions as well as you have search means videos with you, I think, in the NPTEL. So you can look into it and later on you can ask me. Okay. Well, let us start today's session. So I was talking that last year, one student asked me some questions from the week 4, and while I was teaching, so I was trying to discuss that with you. So the first question was why six-fold symmetry is favored over other symmetries. So since Shani, you are not, I think you have not catch upon the previous session. So for you, I am giving you a little bit summary. So what is important? So we have started with extracrystallography in previous weeks. Okay. So in X-ray crystallography, what is very important is that you should have a three-dimensional crystal, right? So that in that crystal, the atoms what are present, they should be like here. This, if you compare this, means if you see my laser pointer in this diagram, if you see each of these atoms, they are present in a particular order, or they are actually repeating itself. Like this is repeating this. Okay, so there is a repetition and there is a order in the crystal. Okay, then only it is if that order is not present, then that will not form a three-dimensional crystal. Okay, so it will be like powdery like substance, or it will be amorphous, and that will be of no use for us. Okay, so in previous sessions we have discussed how we actually made the proteins in our lab. Then from there, how actually crystallizations occur. What are the different techniques for doing that? Okay, and also we have discussed about the different X-ray sources which are actually useful for the diffracting these crystals and ultimately getting the electron densities out of it. And so what is important is that we actually want this, if you consider this as your protein means amino acids, so we want the coordinates of each and every amino acids while refracting the X-ray, by diffracting this crystal with the X-ray. That's why it is X-ray crystallography. So once it is getting diffracted, if I have an image, I will show you. Yes. So here we talked about the different actually, different techniques. So I don't have that exact image, but I will have in today's slide. So what we do is that there is an X-ray source, and from where we diffract these crystals with that X-ray source, and there are different atoms which they actually diffract those X-rays falling on this crystal, and there is a detector which actually detects those diffraction, and we get spots in that detector which are your electron density spots. Okay, now you consider that in this crystal there are so many protein atoms and containing different kinds of amino acids, and each atom, since this diffraction occurs due to the electron density. So there are so many electrons in this crystal. So how can we actually, when we are getting the electron densities, finally that is not our objective, right? We need the three-dimensional structure of your protein. For that, you need to get through the different coordinates associated. You need to know which atom is present in where in the three-dimensional space. For doing that, if you take each and every atom and try to visualize that what is the coordinate, so then it will be very complex for you as well as for the computer, because they will be in a like in a large sea of coordinates from where they have to take. And there are also noises, because in the protein crystal, inside this there is 70 to 80% water. So you don't want the extra diffractions of the water. So for that, there are different filters we will be talking today. And also, if something comes also, you don't want to consider those things. You want to make your calculations easy. For doing that, so sometimes when you have are solving a jigsaw puzzle, so you are solving a puzzle. So first thing what you can do is that you to visualize that puzzle and to find some patterns. So how to solve that jigsaw puzzle? Okay, so if you are successful in doing or searching those patterns, then you can easily solve the whole jigsaw puzzle. Okay, so for that, what is important is that this since these atoms are in a order in a three-dimensional crystal, each and every atom is connected to one another through one kind of symmetry operators. So if it is symmetrical, then only it will form an order, then only it will form a crystal. Okay, so there are different kinds of symmetry operators like mirror, rotation, center of inversion, and glide. Okay, so I have discussed each of these means operators in the last video. So if you look into it in the YouTube, you will find it. While discussing it here, I said that, so you see this rotation screw means one object is connected to another through a rotation screw. Means if you rotate this object 180-degree, you will find another object in the crystal. Okay, so in this is called as the angle or axis of rotation. Okay, so while discussing it, I was telling that among all the folds of rotation, six-fold rotation is mostly favored as compared to the other folds of rotation. Yeah, hello. You cannot see any slides. Yeah, I was presenting. Okay, sorry, sorry. Okay, okay, okay. So thanks for sharing with this, because no one was present earlier, right? You left the meeting, so I forgot to share the slide. Sorry for doing that. Okay, so now you can see, right, the slides. So I was telling that each atom in a crystal is actually associated with one another through some symmetry operators like this mirror, center of inversion, rotation, or glide. So I have described all these symmetry operators in the last video. You can in details what are these symmetry operators. You can look into it. If you have found any doubt, you can actually ask me. But what I said last class is that in this rotation operator, rotation symmetry operator means that if you have one object and you rotate, that means the position of that object to an angle, you will find another object in the crystal. So that is then you can tell that those two objects in your crystal or those two atoms in your crystal is related by a two-fold symmetry. Means it is connected by 180-degree angle. Okay. So if someone tells you that if it is two-fold rotation, then what is n-fold rotation? So the n-fold rotation, it may be five-fold, four-fold, three-fold, six-fold. Okay, so what how to actually visualize that is that you take 360-degree angle and divide it by the number of folds. Right? If it is two-fold, then 360 divided by 2, that is 180-degree. That's why these two objects are connected by 180-degree. Okay, the coordinates of these two objects are connected by 180-degree. Now, if you consider a six-fold rotation, means where each and every object will be connected to one another or related to one another by a 60-degree angle. So, if you find this object and find the coordinates of the object, and if you take a 60-degree, you will find another object. Okay, so those two objects are connected by six-fold rotation. Okay, and these six-fold rotations are mostly found in a crystal. So I actually said that, and I actually asked that why that is so. So that I was discussing today in the first question, that why six-fold symmetry is mostly favorable than other symmetries. Okay, the reason behind that is that if you so for your example what I have taken, so I have changed the slide, right? You can see, right? There are two pictures, one is with pentagon and one other with this. Okay, so in the right hand side I have to give you the example, I have taken two examples, one with the hexagons and one with the pentagon. So you can imagine this hexagon as your six-fold symmetry. Okay, in six corners there are six atoms. In the pentagons, if you can consider that in each corner there are five atoms and they are related to one another by a five-fold symmetry. Now, if you consider this as your unit cell, which consists of all these four atoms, five atoms at the corner, and this is another unit cell which is connected, where each atom is connected by six-fold symmetry. Now, if you want to pack, means pack a lot of these hexagons with one another, you find that there the space between them is very lesser if you compare this with other folds of symmetry. Where whenever you want to pack these unit cells, and this packing is very important to give your three-dimensional crystal a symmetry or an order. If this packing is not efficient, if there are very large spaces or ununiform spaces between these unit cells in the crystal, then your crystal will not be a proper crystal. It will be like amorphous substance or some other form, and that crystal will not be able to diffract and give you that three-dimensional structure. So to maintain the three-dimensional structure, what is very important is that you need to maintain an order, and that order is properly maintained, best maintained with six-fold symmetry. Because if you consider this as, if you draw a circle here in this hexagon, you find that there are very less space which is actually empty. And if you draw a circle here, there are many spaces which are empty. Now circles, you know that if in a space, if the circle have a minimum surface area, if you have something which is circular or spherical, right? So for that also you can take an analogy and tell that this is the reason the packing in the six-fold symmetry is best as compared to the other folds of symmetry. So that is the reason why six-fold symmetry is mostly favored. Now, Shani, if you have any questions in between, you can just stop me and ask me, right? So that will be good. Now, yes. Now, okay, yeah, yeah. Yeah, so that is means one of the reason, because these kind of questions are open-ended. So what people understand is that, so this is actually found in nature. So if you see in nature the crystals, there you will find mostly six-fold symmetries. Okay, because it is mostly symmetrical, more symmetrical than as compared to the others. Then people wondered that why that is so. So one of the reason maybe what people have jotted down is that in six-fold symmetry, the spaces in between each unit cell is very minimal if you compare it with other folds of symmetry. Okay, so that's why, like five-fold symmetry or four-fold symmetry. Okay, so that's why people said that in crystal, for getting a three-dimensional ordered form, you need to minimize these empty spaces. Otherwise it will be more like a fluid, right? So what is the difference between fluid and your so 1 minute, one another one is trying to join. Okay, so so then so for to minimize the space, this six-fold symmetry is more favorable. Okay. So the next question that I we discussed is that why there is a lead coating around the X-ray diffractor. So we discussed last class about the different X-ray diffraction sources. So there I said that in the X-ray diffraction sources where this from this anode this means X-rays are emitted, and while after the from the cathodes the electrons are ejected in means speed of near to the speed of light, and there is no obstruction in between since there is vacuum inside that. So after the x-rays are generated from your anode, so then in the anode there is a lead coating, means outside the x-ray generator there is a lead coating. So why that is so? So the answer to this question is that this lead is an absorber of X-ray. Okay, so if there is any leak while generation of the X-rays, and since you know these X-rays are hazardous to our health and that can cause various diseases, including the cancer. Okay, so means or means it can actually be very harmful for us. Okay, so sometimes what people did previously, this was absent, so many x-ray crystallographers means faced many health issues associated with it. But nowadays, for a precaution measure, since lead is a very good absorber of x-ray, there is a coating around the x-ray diffractor. Okay, so that there is no leakage of x-rays and the hazards can be minimized. So this we will also find while we will discuss the cryo-EM, because in cryo-EM also there are some remnants of these X-rays are also produced. Okay, which can hamper your health. So for there also there is this lead coating. So where we actually left last class is that we were discussing about the x-ray sources, and also the means in the crystal there we discussed that inside the protein crystal you have 70 to 80% water. But now this water is can also diffract these x-rays, and these x-rays will again fall in the x-ray diffraction means in the detector, which will add on noise. But this water is not in a ordered form. That's why we said that the diffraction pad spots or the electron densities for water that you will get, that will be very minimum, because therefore that's why we need a crystal, because in a crystal, since there is a pattern, the intense all the electrons vibrate with one another in a symmetry, or in respect to one another there is some kind of symmetry, due to which you get a larger intensity in the x-ray diffraction pattern. But in water, since it's a fluid, there is no pattern, there is no orderness, you will not get that much. But still there will be some noise in your diffraction pattern. And also, while generating the x-rays, we discussed that there are two kinds of means wavelengths of that is generated from two different energy levels. One is from K to M and another one is K to L, right? So we don't want the K beta. Okay, the higher energy a higher energy emission, means higher energy jump of the electrons from where this wavelength is present in of copper. We don't require that, we only require the K alpha wavelength of the copper. So how to actually get rid of that? For all those, people have used this use of monochromator. Okay, so where you will be actually narrowing your wavelength of your source to a particular form, that there will be a single wavelength. So monochromator means it will emit a light or a wave which have a single wavelength. Okay, so that is the role of this monochromator, and it transmits a mechanically selectable narrow band of wavelengths chosen from a wide range of wavelengths available at the input. So this can actually minimize the this K beta or other noises which can come with your X-rays, because that will actually add on to the noise in the detector. Okay, you require only the K alpha wavelength to come and hit your crystal, so that you get a X-ray diffraction, because the K alpha has the wavelength of 1.5 angstrom, which is similar to the distance between the carbon-carbon single bond. And since we talked about the last class that um, whenever you want something to see by your eyes, the wavelength of the light which will be used, or the wave which will be used to visualize it, that should be proportional, or that should be comparable with the distance between the two atoms of the object that you want to visualize. Okay, so since in the atom we know that the carbon-carbon single bond is around 1.54 angstrom, so we want the X-ray wavelength to be in that wavelength. So only then we can actually get higher resolution images of that crystal. Okay, for that we only require the 1.5 angstrom to go and hit the crystal, and we don't require this k beta, and that can be done by filtering out this wavelength using this monochromator. Sometimes different metal filters are also used, like this nickel foil filter, which can actually obstruct the K beta X-rays. So this answer is also, this is very important with one of your assignment question I have seen. So there the sir has asked that what is used to actually reduce the K beta wavelength in the X-ray means generation. Okay, so that will be your nickel. So we will discuss the week 4 assignment today. So there you will find that one of the question was asked this. Okay, so this is your monochromator, this is your source from where the X-rays are coming, and you want a single wavelength of X-ray to hit your crystal, so that the overall noise is minimized. Another noise is due to what I mentioned earlier, that there are certain heteroatoms or water which can actually, or there can be some fluctuation because of temperature, which can add on to noise to your data. So to minimize those, there are certain filters, and there are two kinds of filters which are found. One is interface filter, another one is your crystal filter. And the demerits is that in the interface filter, the filter is the major drawback of this filter is that the background radiation is still high, and that the transmitted radiation is still not very monochromatic. So there within the interface filter you will have a overall background radiation still high, so the noise by using an interface filter can be minimized, but to a level. But if you use a crystal filter, total crystal filter, then it helps in improving the resolution and also minimize the sample preparation, improve signal to noise ratio. So in a nutshell, the crystal filters are much good if you compare with the interface filters, and it can reduce the noise of your noise of your data minimally. Okay, maximally. So it can give very minimum noise to your data. Okay, so this is your means different types of filter. Now we are looking to, so we have talked about how to reduce the noise in the X-ray generation source, then in between, and then we are going to the detector part. So there are different kinds of detectors, and there are different properties of the detectors upon which different detectors are used in different purposes. Right, so like there are single photon counters which is of the scintillation type, or there can be photographic film which are image plates, like image plates are made by depositing a thin layer of inorganic storage phosphor or a flat base, and X-ray photons excite electrons in the material to higher energy levels. So similar to the means X-ray generation, where these electrons were going from the lower energy level to higher energy level, and then when they are coming back to their ground state, they were emitting the X-ray. So there are certain image plates which are made up of thin layer of an inorganic storage of phosphor or a flat base. Okay, so that X-ray photons excite electrons in the means X-ray photons. The X-rays will hit the image plates and it will excite the electrons, and that electrons will go to the higher energy level, and while they are coming down they will emit some energy that will be amplified by certain systems, by certain computer systems, and that will be recorded. Okay, there are certain area detectors, but what is mostly used is this CCD, that is charge coupled device based detectors, which has a very fast response and a low noise as compared with the other detectors that I have talked here about. Okay, so mostly this kind of CCD detectors are nowadays used. Now there are means when you are talking about the X-ray or any scattering event, you have to consider that there are two kinds of scattering that can happen. One is your elastic scattering, another one is your inelastic scattering. So in inelastic scattering, what is happening is that the waves that are being generated by your inelastic scattering, they are not in a phase with one another. Sometimes means that this, if you add this constructive means this if you use this inelastic scattering for while actually diffracting your crystal, you will not be finding any X-ray diffraction, or it will not give any diffraction. Okay, so they are incoherent scattering, so they will be like a noise. So the Compton scattering will be mostly by the water molecules which are not in an order. Okay, so they will be doing inelastic incoherent scattering, and they will actually add up to the noise. Whereas in the crystal, in a three-dimensional crystal which has a particular order, if you hit the X-rays with in that crystal, if it is a proper crystal, it will actually perform a Rayleigh scattering. And the Rayleigh scattering is a elastic scattering, and it will lead to a X-ray diffraction pattern, which will actually help us to solve the three-dimensional structure of the protein. So what is very important is that for to form crystal is that, see, this is the use of the orderness of the crystal. So if there is no orderness, there will not be this elastic scattering, and you will not get any X-ray diffraction pattern. Rather, you will get noises just like your water molecules inside your crystal. Now, another important thing while we were discussing last class is that there was constructive and destructive interference between two waves of the X-rays. Okay, so if the two waves are in constructive interference, means that the phases of those two waves are similar. They are in phase with one another. Okay, then it is said to be constructive. Means the draw of that wave is similar at a particular time or at a particular wavelength. This draw is coming there in a particular time period. Both of them has a similar draw and similar crest. So when you add on these two waves, it will give you a higher amplitude wave. So what we see in the X-ray diffraction or in the detector is this constructive interference. So that's why you require your atoms to be in an order and to be in a phase, so that you get a constructive interference out of it. If you get a destructive interference, it will not give any electron density in your detector. So for to avoid that, we actually require that three-dimensional ordered crystal. Okay, so this is what was left out in the last class, and then what we have is that, so now we know that we require an X-ray source, we require something a crystal and a detector. So this is the basic idea that we have. Now, since in the last class, if you see my previous video, we were discussing that these crystals, these are made up of different means unit cells, and that unit cells have different kinds of means lattices. Like means for a particular crystal, it will have there is some kind of system. Means space groups are there, point groups are there, which actually make your asymmetric unit, and that asymmetric unit makes your unit cell. And your unit cell, the shape of the your unit cells are important to determine in which lattice system your crystal is present. Okay, so it is like each and every crystal will follow a particular lattice system. For there we discussed about the 14 Bravais lattices, and so the all those face centered, body centered, eye centered, and all those hexagonal, monoclinic, and this all those lattice systems we actually discussed. So those will be present. Okay. Now there will be so the first thing is that when you will get those lattice systems, you remember that each and every unit cell is defined by the three vectors, basis vectors A, B, and C, and the three angles alpha, beta, and gamma. So now if you think that this A, B, and C, these can go from 0 to infinity, right? So if this is a very large crystal, then the diffraction patterns that if you want to compute by just like A, B, and C, then you will have points from 0 to infinity, and that is a huge collection of points, right? So but you want to minimize that means your data set, and for doing that what we do is that your diffraction patterns, this that you get, is not in a real space. So your crystal lattice, so if you consider, so this is a little bit difficult to understand, so try to understand like that you and your shadow. So you is present when something, your light is coming and hitting you, and there is a shadow in the wall. So the coordinates of yours is can be visualized in the real space because you are present in reality, right? But the shadow which is being formed, that is for an analogy I am telling that that is not in a real space, that is present in some other space. Okay, so that is what happening in this crystal structure. The diffraction pattern that you have in that detector, so that is not present in a real space, it is present in a reciprocal space. So what I mean by reciprocal space, in the last class means so let me revise because many of you were absent. So if you consider, we actually talked about the different Miller indices. Means just like these different basis vector that A, B, C that can define your crystal in a real space, you need some Cartesian coordinates to define any object in your reciprocal space, and you know that your diffraction pattern that you are getting, that is a reciprocal space, right? To get information out of it, you need certain coordinate system, so that is actually given by your Miller indices. So what are Miller indices? First let us see. So if you connect, these are all these lines that you see, these points are your atoms, or imagine these as your objects inside the crystal. If you draw lines through this means objects, you will, if you imagine this in a three dimension, that is x, y, and z, or a, b, c means basis vector in a crystal, each of this is a plane. Okay, so these planes sometimes this can be similar, same as your axis vector. Means what I am saying you that 1 minute, let me take the pen. So there are three basis vector A, B, and C, right? So there are three vectors on the basis of which you can actually define each and every coordinate of your atom in a real space, right? Now there are different atoms present, means there are different atoms present organized here and there, right? So some of them will be like parallel to your axes, right? Like this. But some of them, like if you connect these 2 points, this lattice plane, the plane which will be forming by these two points, this will cut your A and C axis at some points. Okay, and also your B axis, means if you, this is I am drawing it in the AC plane. Okay, but your crystal is 3D. So imagine this scenario in a 3D space. So there are different planes which will cut your these three basis vector at some region. Okay, if you have a plane which is at an angle with this A, B, and C, So that plane means the plane that is cutting at different points. So this is one point. Here it will cut at one point. Here it will cut at one point. If you draw a plane, okay, in a 3D direction. So this will cut and there will be some intercepts. So if you take the inverse of these intercepts, you can find the Miller indices. So what is the advantage of this Miller indices? So remember what I said, that in a crystal, in a real space, what is the difficulty is that this A value in a real space, it can go to 0 to infinity, right? But in Miller indices, what you are doing is that you are taking a reciprocal, means wherever this intercept is there, so this intercept you are taking a reciprocal, right? So you are taking 1 by A. So this can give you a value that we considered as H. Okay. Then you are taking 1 by B. So that is related to K. And then again 1 by C, that is related to L. So this HKL, since if you have infinity or if you have very large value of A, then also you are restricting your data set to some small number of points, because this is always a reciprocal. So the here from here also you can actually visualize is that sometimes you will see that if you take in a chemistry, if you work in a chemistry laboratory, you will see that the smaller salt crystals, they have very high resolution. Their images are of a very high resolution. But your protein crystals, since they are very large and the distance between each atoms are very large. Okay, so this due to which this, if it is very large, the HKL is very small. So due to this, because this HKL are very becoming very small as the crystal is becoming larger, the resolution of those larger crystals are less as compared to your smaller crystals. Because if you have a smaller crystal, this A value will be small, means the distance between two atoms will be small, then your H value will be higher. So you will get a larger reciprocal lattice of those smaller real crystal. Okay, that's why smaller objects can be actually having higher resolution, whereas very high larger crystals like that of protein, the resolution limit, there is a resolution limit to which we can go. Okay, so this is the rough idea of Miller indices. So sometimes in the question, they will give you this A, B, and C value or X, Y, Z value, which is similar to your A, B, C. Okay, if you find X, Y, Z, that is your means in X axis. What is the means? What is the intercept? So first what we need to do, so how to find Miller indices. So if they give x y z values, x y z intercept values, okay, first what we need to do is determine the intercepts of a plane along the x, y and z in terms of their lattice constants a, b and c. So that we have done here. So this is, so if you find this is a, so a means x, right? So that is 2, means 2 units. After 2 units from 0, their plane is cutting the x axis. So that's why the intercept here is 2. Now the plane in y axis, it is cutting at 3, right? So the intercept of y axis here is 3, and again in the z axis there is 2, so that we have written here is 2. So first, whenever a plane is given to you, you need to find that where this plane is cutting the 3 axes, and you need to get out these values. Okay, once you determine these values, you determine the reciprocal of these values, means just make inverse 1 by 2, 1 by 3, 1 by 2. Okay, now what you do, this 2, 3 and 2, the denominator, you take the LCM, LCF. Okay, and this LCM, if you take LCM, if you take, that is 2, 3 and 2, you have 6. Now you multiply that LCM with each of these values. Means 6 multiplied by half. 6 multiplied by 1 by 3, and 6 multiplied by 1 by 2. That is your 3, 2 and 3. This is your H, K and L. So this is, you are getting Miller indices from the intercepts of a real lattice plane cutting your real axis. Okay, so this is how you actually found the Miller indices. This is clear. Means how you can find the Miller indices. If it is not, then I guess it is clear, right? So it is very important, right? You can be asked in the assignments as well as in your exam. Okay, so it should be very clear. Okay, so from here we are actually going in this slide, in today's slide. Okay, so here what we know now, that our crystal is in the real space, whereas our diffraction pattern, this is in the reciprocal space. So in the diffraction pattern, from the diffraction pattern you get something called as structure factor. Okay, so you imagine something like if you take a lot of sine waves and you combine them with one another, because individually the sine wave amplitude was less, but when you take constructive sine waves, just the way I showed you in the Rayleigh scattering, if you take constructive sine waves and add them together, you will find a final wave, right? Which will have a higher intensity, right? Similar to that, so that what you are doing there is a Fourier transform. Okay, so that is a simple way to imagine. So if you combine a lot of sine waves and combine it into a larger sine wave, means add all those waves to a final wave, you are doing a Fourier transform. Now if you want to revert back, means what are the components, all the sine waves that you used, you need to identify each and every sine wave from the final sine wave. So you need to do a inverse Fourier transform. Okay, you need to perform a mathematical function on that final sine wave, and you need to invert, means perform an inverse Fourier transform to get into an individual components. Okay, so what we do here is that you get diffraction pattern, but you don't want that diffraction pattern. You want the electron density in your computer. So in your computer has this algorithm where they will actually take this diffraction pattern which is coming from the diffraction of your crystal, and this diffraction patterns are forming in the reciprocal space. So from there your computer has this all these calculations that we do HKL manually. The software can do it, means by computationally they have the algorithm and they will calculate your HKL and made your means reciprocal space where you will have this diffraction pattern, and for each and every spots you will have a structure factor. So what is the mathematical significance of structure factor, we are coming. But you imagine something that what is electron density in your real space, that is structure factor in the reciprocal space. So if you, structure factor is like the Fourier transform of your electron density, but you don't want the structure factor to solve the your crystal three-dimensional shape. You want the electron density, but what you are getting from the diffraction pattern is the structure factor. So then what people do is, what not people, the computer do, the computer actually applies an inverse Fourier transform in that structure factor equation to get your electron density. And these electron densities you can actually see in your softwares like the spots. Okay, so this is what is done by the software or the computer. So this is the way you, this is the means logic that is going in this structure determination. Now, the structure factors are also denoted by F. Okay, so this structure factor square is your intensity. So this structure factor you can actually make an analogy with the amplitude. Okay, so the amplitude square is your intensity. So from this structure factor equation you actually get the idea of the intensity, but the information about phase is lost. So if you want to detect any wave or any coordinates of any wave, you have two important things in a space. Okay, one is your amplitude, another one is your phase. Okay, but from the structure factor you are only getting the intensity, so you will get these bright spots, okay, dark spots. But how these dark spots are connected to one another, that information is given by your phase, but that phase is lost while calculation in this inverse, means generally lost while if you take a simple procedure, if you follow this simple procedure, you will not find the phase information. That is the famous phase problem. So we will look into is mathematically as well. Okay, why these phases are lost. Hold on for some minutes, we will discuss that. Okay, so all these diffractions for, see, these are present in the reciprocal lattice. So it will have all these Miller indices. Instead of your A, B, C, you have coordinates of H, K, L. This will be actually your Miller indices. Right. So what is this plate that you see? These are known as Mar plates. We will be coming to this. Okay. So first thing is that, so this is to summary what we have talked till now is that there is a diffraction pattern. So this is an X-ray source. There is a primary beam. It is coming to a crystal. It is diffracting and it is falling in a detector. And these are the diffracted beams which will form some densities, and this diffraction is occurring in a reciprocal space from where we will get the structure factor. And from the structure factor equation, if we do an inverse Fourier transform, you will get your electron densities. Okay, that we actually know till now. Now this diffraction intensity, this means the how much this structure factor value is increasing or not, that is proportional to the number of unit cells in the crystal. So if a crystal has a large number of unit cells, the diffraction intensity will be higher. Okay, now also what we learned is that for getting a diffraction, there should be a constructive interference and a Rayleigh scattering, and this constructive interference will only occur if the X-rays follow this Bragg's law. Means in a crystal, if it is following this Bragg's law, that is 2d sine theta equals to n lambda, then we derived this Bragg's law last class, so you can go to the last class video and check that how why this Bragg's law is important for the constructive interference. So now means now whenever you get a crystal, so you first thing I said that getting a crystal is very difficult. Sometimes you getting a three-dimensional ordered crystal, it takes years. Okay, but once you get that crystal, still not, it is not solving your whole problem. That crystal, I have repeatedly said that the your crystal should be in a order. Okay, so that order, if it is not ordered, if there is some kind of defects in your crystal, and if you use that crystal for getting the three-dimensional structure, you will end up with your erroneous data or a fuzzy data. The resolution will not be that much good. So for that, whether your crystal at the initial phase only, whether your crystal is defective or not, you can take that crystal and take a initial diffraction pattern. You don't analyze anything, you do not do any computer analysis, anything. You just are checking whether this crystal that you have made, that can give you better resolution three-dimensional structure or not. If that can't give you, then doing that process and everything, that is also take time taking. So why will you waste resource and time? So to minimize that, what people do is that you take the crystal and diffract it by a home source and take the electron density spots. In the electron density spots, if you see that these electron density spots will have certain Miller indices, and from the Miller indices you can actually there is this algorithm in this software which will see that how these Miller indices are connected to one another and detect that whether this system is triclinic, monoclinic, or everything. Okay, so that it will actually denote. Okay, someone again wants to join. Okay, so so what we were telling is that initial diffraction of your crystal will give you some diffraction pattern. From by looking into this diffraction pattern, you can tell that whether your crystal is actually ordered or not, or if it has any disorder or not. Like if the crystal which will give you a higher resolution structure should be in a perfect order, like one should be arranged, there should be a serial arrangement, there should be a periodicity, but there can be some disorders in your crystal as well, like rotational disorder means some of the plane, one of the plane of the crystal is is not aligned properly with the another subsequent plane. Okay, and it is actually rotating. The relation between these two planes are by a rotation. Okay, so if you now try to diffract, since you see these two horses picture, there is some the if you think this in a three-dimensional space, and if you try to see from the front, you will see little bit behind of the horse behind in the second plane. There something will be seen because this is not in a perfect order, due to which the diffraction spots which you should get from this perfect order, that this is similar to this is similar to this, so the diffraction spot that you will get, that will be very distinct. But here, since there is a defect, there is a rotation, if you pass a X-ray through this plane, since the subsequent plane are not parallel to this plane, they are in an angle, the diffraction you get is a fuzzy diffraction. You will not get this kind of spots. Okay, and there can be some translational disorder. Means here it was rotational and there can be translational. Means this second plane is something higher or lower as compared to the previous plane. So this all these disorders you can actually directly see whether by diffracting your crystal by a home source. Now when I discussed you about the means about a crystal, I said you that means X-ray crystallography. I said you that the crystal is mounted on a goniometer head, and that goniometer head can be rotated in different angles because you want to collect the data of your crystals, means diffraction data, from different frames. And if you are actually only getting diffraction pattern from a single frame, it will give you a limited information. You want the data from different frames in the crystal. Okay, so for doing that, you need to rotate your crystals. Now, if you are using a home source, the X-ray diffraction intensity is very less. To get the means X-ray initial X-ray means intensity, the source, the home source that will generate, that is less as compared to synchrotron. So when you are actually hitting that X-ray in a single plane, you need to hold the crystal there for few seconds. Means you need to have a time, you need to give a time for the X-ray to do the diffraction, because the intensity of the initial intensity of the X-ray is lower for the home source. This problem will not occur for the synchrotron. So when we were discussing synchrotron, I said you that in synchrotron you can get the X-ray diffraction pattern very faster. Why that is so? Because this delay time is absent there. You can get the diffraction pattern in very minimum time because the initial intensity of the X-ray is very higher for synchrotron source. But for in-home source, it is lower. So there will be always a delay time or readout time, that is 5 to 7 minutes. After 5 to 7 minutes, you again rotate the crystal by an angle. So what we do is that by 0.5 degree, the angle, the crystal is rotated, and you take the data in each frame by keeping that angle 5 to 7 minutes. Then again you rotate take the data, means that you don't rotate. There is it is these are robotized, and you tell the means goniometer you set in such a way that this amount of time, after this amount of time, this will rotate its head by this angle. Okay, so now you see that if you have 600 reading frames and you take this much amount of time, you need 3500 minutes, and this is a very less time that I am telling you. So to minimize that, so what people do, so the nutshell of this slide's discussion is that if you get some crystal and you have the opportunity to send it to a higher X-ray intensity source like the synchrotron, you just don't pick that crystal and send it to abroad, because in India we don't have synchrotron. You need to send it to France, Italy, Germany, they have the synchrotron to get high resolution structure. But if your crystal have certain defects and you are sending it to the means higher means this abroad, and that will be the waste of your money as well as your resource. So for doing that, what you do is that first you need to check whether your crystal is good or fit for a better resolution image or not. You have some in-home source, you take that crystal and take a initial diffraction pattern. Look into it whether you are getting this kind of distinct spots or not. If you are not getting, if you are getting like fuzzy patterns, then you have some problem with your crystal. You need to prepare and again the crystal, or you need to choose another crystal. This that fuzzy pattern will be hampering your data. When you are sure, 100% sure with the in-home source that your crystal is better for a higher resolution image, then only you send it for the abroad or in a higher synchrotron source. Okay, so this brings me to this smart plate analysis. Okay, so once you get, now you have sent your crystal, you have looked into that your crystal is fit for your good resolution. Now you have sent it to the means to a source where it has higher intensity X-ray source, and it gave you some diffraction patterns and some electron densities. It did some Fourier transform and it gave you this electron densities. So how to visualize it? So to visualize it, there is this MOSFilm software. Okay, you can use this MOSFilm software, and this is your this round plate that you see. This is your MAR plate. Okay, so in this MAR plate, the more you go away from this center, okay, if you go away from this center, let me take the laser. So if you go away from the center to the periphery of this smart plate, the resolution increases. Means the points, the electron densities that you will find here, the resolution of those atoms in the final image will be much higher as compared to what you find here. But you see that since your protein crystals are very larger, we don't get very high resolution. It means to this level, you see there is no spots. Mostly these spots are present around this center. Okay, so here you will get this, and there you see there is a small hole in between. What is this hole? This is a beam stopper. Means when you are actually hitting the X-ray to your crystal, you if it hits your crystal at a zero angle, directly hits your crystal, then it will heat up your crystal, destroy your crystal, or it can hamper the orderness of the crystal because it will increase the energy. So that's why we don't want the direct X-ray to hit your crystal. That's why there is some kind of metal stopper or a beam stopper, which is a metal disc like shape, which can actually, which is there is a knob attached to a metal rod. Okay, just like you see here. So this is a axis detector. This is different from this one mud plate, where you will have square. This image is square. Here you have circular. Okay, so this is a different kinds of, there are different kinds of detectors. Okay, so this is a Raxis detector which use this Rigaku generator for the in-harm source that I discussed in the previous lectures. Okay, in discussions. So here you see, since there is a beam stopper in this region, nothing has been diffracted, because if this was also diffracted, then this here the crystal, maybe the orderness of the crystal would have compromised. So that's why you get the sum of the data which was present here, that actually got lost. Okay, so that's why now people, how to design this beam stopper, that is also in research, because means you need to minimize the area of beam stopper so that you will get this lost spots that you see here in this right hand side, that can be minimized. Okay, so once you get these spots in your mark plate, now what you need to do, means what this software do, is that it will take this each points which is present here and try to find any pattern. So since I said you that in the unit cell they follow different Bavius lattices, and each Bavius lattices, we know that in the triclinic system what are the different space groups will be present of the unit cell. Okay, so it will scan through all the patterns by looking into these points in the space, and it will say you that this crystal, for example, is following a triclinic system, and with this space group is present here. These space groups are present here. Okay, so if you are not aware about space groups, points groups, you can look into previous lectures where I have discussed that what are point groups, what are space groups. Means with this symmetry operators, if you actually link a subsystem with one another, it will actually, this symmetry operators will form a space group. Okay, means in the space some atoms are in a group which can be actually connected with one another with certain symmetry operators. So that in a nutshell I can tell you, but for details you can see the previous videos. So in the there I have mentioned what is the space group. So what this software do first is that it will determine what is your system, crystal system, and it will tell you that where, in which space groups are present here. Then what it will do, it will find the ABC just like that we did manually, and try to draw lattice planes through that ABC, and try to find out that intercepts. Once it find out the intercepts of the lattice planes, just we did last, means few minutes ago, how we find the Miller indices, the same thing it will be doing here computationally. It takes the intercepts, it takes the reciprocal, it takes the LCM of it, multiply it, then it gets the Miller indices. So each point will have a Miller index. Okay, so that is known as indexing. Then what you need to do is that you need to, there are different frames in which you will take this data, right? And in each frame you are looking in a same object. So for the same object you need to take data from different frames, and then you need to integrate those data so that you can find a final X-ray diffraction pattern. So that is under this integration. Finally, what you need to do is that you need to merge all those images by actually minimizing the noise. Means there will be some spots which will be adding to your noise. So you need to refine this electron density map with the help of certain, means scaling factors. Okay, so there are certain scaling factors which will be actually utilized to actually tell you whether those spots are means they are actually eligible for giving you a proper information about your 3D structure, or they add up to your noise. So there may be thermal noise, there may be some noise due to the distortions, and each and everything that you need to screen before the final data processing, that comes under the merging process. Okay, so this is the software sometimes what means in this software, what it happens is that this when you give your marplate, it will be showing you some spots by itself. Means it will have that means software knowledge that here this electron density spots are present. But sometimes what will happen is that the actual electron density pattern and the spots made by the software, they are not aligning properly. Then you need to zoom in this region and try to align those two spots means properly. Okay, then only you can actually get the proper information. And sometimes you can see these red spots. Red spots means over intensities, means this software, the diffractometer, cannot detect these intensities because these are too much for the limit of this means software. Okay, so that all those information you can actually get, and this is what I discussed in the previous slide, that how you to know that whether your crystal is properly ordered or not. You need to actually take that crystal, use some arbitrary angles, 0 degree, 90 degree, 180 degree, because if you take all those angles 0.5 degree, you need a lot of time, and this will not give you a final data because you will be sending it to the synchrotron. For that, you take some random angles like 0 degree, 90 degree, 180 degree, and take the extra diffraction pattern and try to visualize in it whether it is defected or it is everything is fine or not. Means whether these rotational translational disorders are present or it is absent. Okay, so this is all what we discussed by theoretical knowledge. Now, if someone wants to visualize this in mathematical form, means why phases are lost, what is this structure factor, how this scattering is occurring, what is the equation on which this, I told you this diffraction pattern will have a structure factor. You need to do a inverse Fourier transform to get these electron densities. How this software is doing that? So to know that, you need to go into mathematics. Okay, so I will continue this from this to means today if I get some time. But what I will do now first is that, since I need to discuss with you the assignments, right? So this is the week 4 assignments. So first let me discuss the week 4 assignment solutions. Then if I have time, then we can go to that mathematical part, or we can do that in the next class. Okay, so so again, I don't know whether I will be having some time for the practice questions or not, but these slides, everything is being shared to you in the Google Drive. So the Google Drive link has been shared with you through NPTEL in your means in the course. If you go in the NPTEL, in the announcements, they have shared with you this Google Drive link, and also they have said you about the video lectures. So all these materials in the Google Drive, I am uploading these materials, these slides, as well as your videos, the videos that I am making. This session is being recorded. Okay, so that will be also uploaded in the Google Drive, and also there is a YouTube channel. If you can go there, that is being made by me, that is NPTEL BT10 structural biology. If you go there, you can find the previous videos as well. So you can solve these questions at home. If I have time, I will do that. First let me solve the questions of assignment 4. And before that, the if you guys have any doubt, you can ask me. Means anything from today, or any doubt, because some of you have joined later on. So any doubt, or this so the people who have left, means who have not listened to the whole means 2 hour session, or they have means missed out some part, they can actually look into the video recording and can ask me questions in the next class as well. But today, if you want to ask something, you can ask me. Otherwise, we can actually discuss the assignments first. Any questions? Anything which I need to clarify? I am audible, right? Okay, okay, okay. Then if you have any questions, others, you can write in the chat box. Let me discuss the assignment. Then in the end, I will see if you have any questions. I will answer. Okay. So so the first one is the this is your week 4 assignment. So the solutions are already given, but just this is my responsibility to discuss with you about the answers. So so the first, these are the first 5 questions, and we will go each and every question one by one. And in any a solution, if you have any doubt, please do ask me in between. Okay. So the first question was wavelength suitable for XRD application ranges for. So since from the first day when we actually discussed about X-ray crystallography, I have been telling you that the why we have chosen X-rays, because the X-rays have the wavelength in the angstrom region, and the distance between two atoms in the means in a molecule, that is like C alpha, means if you compare the carbon atom, the carbon carbon single bond distance is 1.54 angstrom, and that actually matches with the wavelength of the X-ray. So here, so you don't need to remember the exact value also to answer this question, because sir has given you in different units, because he also only wants to know whether you actually have the overall idea, because these X-rays need to be in a angstrom. So by just looking into the unit, you can come to the correct answer. So the answer to this question in your assignment 4 first question is this B option, zero point 0.5 to 2.5 angstrom. Okay. Now, in the second question, in small molecule crystallography, which of the following source is used? So, when we were discussing this X-ray production, I discussed with you that this anode is made up of generally two metals. One is your copper, another one is your molybdenum, because they are DD transition elements, and these two means metals. There, this energy gap is such that when the emission occurs, the emitted wavelength is similar to the X-ray wavelength, and that is similar to the space between two atoms. So that's why we have chosen two, copper or molybdenum. But the wavelength from the molybdenum means your anode is this zero point 0.7 angstrom, which is much smaller or half than your copper. So this MO is mostly used for small molecule crystal, but the copper is used for mostly your larger crystals like protein crystals. So this we already discussed in our last class. So the question is, in small molecule crystallography, which one of the following source is used? So that will be your option D. So this is also very easy question. Now K to M shell transition of an electron generates which of the following emission? So there only, when we were discussing the X-ray source, how these X-rays are generated, we said that there are two kind of transition occur, that is K to L or K to M. Now these L vibrational states are far away from one another, but in M they are much closer to one another. So there is only one absorption means emission, that is this K beta wavelength, whereas for your alpha you have K alpha 1 and K alpha 2. So to get a average of this K alpha 1 and K alpha 2, that will give you the 1.54 angstrom. Okay, but in this question it is asking K to M shell transition, not K to L. So K to M shell transition generates your K beta. So this you don't require, and this you actually get rid of by the nickel filters that we discussed today, right? So the answer to your question will be C. Then which of the following is not a symmetry operation? Now, since we start today the class with the symmetry operations, I said you about mirror and center of inversion, rotation, and glide. So I described all these symmetry operators in the last class. So you can go there and watch. But if you see here, inversion is a symmetry operator. So I have already discussed with you. Rotation is also a symmetry operator. Diffraction is not a symmetry operator. Translation is again a symmetry, because here you are in the glide, you are translating first and then reflecting. Okay, so translation is also a symmetry operation, but diffraction is the phenomena. So this is also very easy. So the answer to this question is D. Right. Then which of the following represent the two-fold rotation symmetry? Now the all the options that is given solid square, if you see, it is not twofold, it is a six-fold rotation symmetry it has. Okay, solid hexagon will also have that. Solid pentagon, if you see, pentagon will have five-fold symmetry. Hexagon again six-fold symmetry. Okay, this is only the solid oval. If you have an oval like this, so sorry, this is not an oval. Okay, so this is somewhat like an oval. So don't consider this horn. So if this is the oval and there are two objects, so these two objects are connected here by a two-fold rotation axis. Because if you draw here, this is connected by 180 degree to the two atoms, right? So two-fold rotation symmetry is been displayed by this solid oval. Others have higher fold of rotation symmetries. Okay, so the answer to this question will be your A, right? Then the next five questions. So the first one is symmetry operation of glide plane. As I said, you need to first translate or reflect, then reflect, or you need to first reflect along a mirror, then translate. That is your glide. Okay, means if you have said by 2 by A, means first you need to first rotate the two-fold rotation, have a glide along A direction. Along A direction you need to first take mirror and then move by 1 by 1 by means 1 by 2 distance. Okay, so that information, so all this actually we discussed in the last class. So if you go there, you will be actually looking into it. Okay, so the answer to this question here is the rotation, that is your reflection, sorry, your reflection plus your translation. That is the answer will be C. Right, so you are reflecting along a mirror then rotating, or rotating sorry, you are reflecting then translating, or you can translate and then reflect. Okay, so the answer to this question will be C. Then a unit cell has a shape of 120-degree rhombic prism. This unit cell will have which of the following crystal system? So if you want to see the crystal system, you need to remember this table. So you see the only the angle 120 degree is for hexagonal crystal system. So your answer to the question will be B. Okay, so this table better to remember it. So remembering this is easy. Like for cubic, you write like A equal to B equal to C, alpha equal to beta equal to gamma equal to 90 degree. So here everything is equal in cubic. Now if you write tetragonal, you cut 1, means B is not equal to C, but still your angle here is equal. That is alpha, beta and gamma. That is a tetragonal. Okay. If it is orthorhombic, then now cut another. That is A is not equal to B. So first we are actually cutting the axis vector. Cutting means they are not equal. We are making not equal of the axis vector, keeping the angles constant 90 degree. So in this pattern, first is cubic, then tetragonal, then orthorhombic. CTO. Okay, CTO. Then now what we are doing, we have this axis vector equal first, and we are cutting the angle. That is the gamma is not equal to 90 degree, but alpha is equals to beta equals to gamma, but they are not 90 degree. Okay, so this is the rhombohedral. Then if you cut C, and if you have alpha and beta 90 but gamma is not 90, that is 120, that is your hexagonal, which is the answer to this question. Now if you're monoclinic, means all of these three is cut and alpha is equal to gamma, but it is not equal to beta, then it is monoclinic. And if it is nothing is equal, means A is not equal to B equals not equal to C, alpha is not equal to beta, not equal to gamma, then that is your triclinic. So triclinic and cubic is very easy to remember, because here everything is equal, here everything is not equal, right? So it is CTO, then RHMT. Okay, so the way everyone has their own way, so I remember like this. Okay, so this is the way you can remember. So the answer to this question is B. Which of the following filter is used to reduce K beta X-rays from copper anode? Okay, so I mentioned today that nickel fault filters are given so that you do not get this K beta value, that is 1.34 angstrom, because you only require this K alpha and you require that to be monochromatic. So the answer to the question here is A, that is nickel. Then which of the following metal is used in liquid state in X-ray sources? So if you see the X-ray sources, so one is your copper here, another one is your gallium. Okay, sodium, liquid sodium and liquid mercury will not give your X-ray source if you make so that will not give the X-rays, right? So gallium can do that, but the exception is, you remember that gallium is present in a liquid state. The copper is present in a solid state. It's a metal, right? So the answer to this question will be the option D, that is gallium. And the final question, that is, generally protein crystals have a big lattice, so the spots obtained for such crystals are. Okay, so this is related to your Miller indices. So I said that the larger the lattice, okay, and so the spots will be very, yes, yes, someone wants to ask something. Hello, is there any question? Okay, no. So yeah, if you have any question, you can just ask me. The thing is that I was telling that the larger the distance between these two atoms, the actually in the diffraction spots, means the protein crystals have a big lattice, so the spots obtained from such crystals. So the diffraction spots, since they are in a reciprocal space, the spots will be, the distance between them will be very close, because you remember this Miller indices are reciprocal of your ABC. So if ABC is more, HKL will be less, right? So in your X-ray diffraction pattern, the spots will be much closer to one another. This will actually lead to the lower resolution, because I said that for protein crystals you actually get lower resolution as compared to salt crystals, because in salt crystals in real space the two atoms are very close to one another. So that will make in the reciprocal space, in the diffraction pattern you get, there the spots, the distance between them will be very high, so you can distinctly determine the coordinate of each spot. So that will actually increase the resolution. So here the answer to this question will be that the points will be very close to one another. That is, the answer is C, option C. Okay, so this is the things of your assignment 4. So if you want to ask me something, you can ask me about assignment 4, right? If you don't, then we can actually look into this mathematical form little bit. Okay, means what is scattering factor, why we are telling that this will go to the reciprocal space, and how from the reciprocal space the Fourier transform is done, what is the equation. Okay, all these things we will learn in a mathematical form, but I will tell you a story, mostly a story, because if you go much into the mathematics, sometimes biologists are afraid of mathematics. Okay, so for many of them this X-ray crystallography becomes difficult, but it is not that much difficult. You don't need to know that much level of maths, because these things nowadays are available in software. You can actually, the software will do it for you. You what you need to do is to how to run the software. Okay. But since if you want to make better softwares from today's, so then you need to know how these softwares are made and on what basis, what is the logic behind that, what is the physics behind that. Okay. So for that, this is important. So do you guys have any question or shall I continue? Any questions? You guys are understanding? No question. Okay. So the things that I have talked you, so it's clear, right? Means, am I able to convey my message to you, or you have any suggestions that I can better myself? I am also a student, so feel free to tell me. Okay. Okay. So I hope you are understanding the things. Yeah. Yeah. Some of you want to tell something. Okay. 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 Thanks for the compliment. So yeah. So then I can continue a little bit. Yeah. Yeah. Hmm. Mhm. Okay. Mhm. Okay. Okay. How many assignments have you missed? So each assignments contains some marks, right? 1 minute. Let me go there. So I think you will be able to cover up because there are many. So you have up to week 6, you have the assignments, right? So there are 6 assignments, and then you have the exam. So the main exam is carrying the means larger part of marks. So assignments, if you have missed 3, then also means you can manage it by doing good in the next assignments as well as your in your exam. Okay. Better what you can do is that you can write this to the NPTEL team by telling that that you have we were not aware about that, but I don't think they will consider. But still, you can try. Means there is nothing, no money for trying, right? So yeah. So you can ask them that I was not able to means I didn't know about this, so I didn't check because of my exams going on, so can I redo that? But they will not allow because these answers are already given, right? Yeah, so I think better is that don't worry much about these 3 assignments. So the marks that you have lost, that is gone, right? So that can be reverted back. But the marks that are coming means do the assignment sincerely, follow the lectures, and these assignments are very easy. So I think that in next 3 weeks you will score full. So many people may have missed, right, your means these assignments. Okay, so that doesn't matter because final exam will be you of your larger amount of marks, and finally the grades and everything. I actually don't know how these grades are given because that is handled by sir and NPTEL. So that is, I think that that is basis on relative, maybe relative means the one who has get highest, then there will be some range on which you will be graded. I don't know, but I am just guessing that. But yeah, but what I know is that this will not matter much. Okay, you just concentrate on future assignments and also your means final exam, that will be carrying the maximum part. Okay, so that will be fine. Okay. 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 So yeah. So then, uh, so we can little bit discuss about this since we have time, 20 minutes. So yeah, what I was discussing with you previously is that the crystals are there. In the crystals, the X-rays are coming. It is diffracting and falling in the diffractometer. or sorry, falling in the your detector, right? Now, if you think like that, means what is inside the crystal? So inside the crystal, there are something called as unit cells that we have discussed. Okay. So that consider okay, unit cells are there. Then what is inside the unit cell? So the inside the unit cells you have atoms, right? So unit cells are made up of certain atoms. Okay, so that is fine. Now, if you go much deeper, that ask that why diffraction is happening, what is there which is actually diffracting. Okay, then you get into that. Okay, inside the atoms there are certain electrons which are actually scattering these X-rays. Okay, so we will go to that level, means we will go from the small to the large crystal, means we will go from the electrons to the crystal level and try to mathematically define this diffraction. Okay, means from the electron level to the crystal level, how this diffraction is occurring, the physics behind that we will understand. Okay, so now first you consider we are in the electron level. Okay, so you see my laser pointer. So there are two electrons inside an atom. So for example, electron 1 and electron 2, and they are in a space. If you take a Cartesian coordinate here, this E1, if you take an origin at E1, then you want to take relation with E2. This E1 and E2 is separated by a distance of r, and this r is a vector here. Okay, that's why it is denoted by this bold. Okay, bold letter. Now this r vector, this is the positional vector. This is connecting your E1 position with E2. Now remember, when this is a coherent or a monochromatic wave, which will have an initial scattering limit, that is this s naught, initial scattering factor. This is this s naught, and this is your after the diffraction scattering factor, which is denoted by scat s. Okay, so this is similar to, if you imagine the in the this is, if you take the magnitude of it, this is similar to the amplitude of this wave that is coming. This is the initial, this is the final. Now when it is coming, since this is denoted by single arrow, because these two should be similar, because we are using a monochromator. Now when it is coming with the X-ray beam which will be hitting this E2, and the X-ray beam which will be hitting this E1, this X-ray beam which will be hitting to E2 will have to cover an extra path, that is P and Q, as compared to your E1. With E1, this is like this, and E2 it has to cover this extra path. Now if this extra path that this is covering, this is not going into a relation where these two waves are constructively interfering with one another, then you will not get a electron density. So it is very important that this electron extra path that this is carrying out, this X-rays, the phase between these two waves remain constant. Okay, so for that, for all those, now let us look into this, how much extra path this has to cover, that is P plus Q. Now this lambda is the wavelength of your X-ray. Now if you want to define P by simple trigonometry, you can find that this P is lambda dot r, means lambda magnitude of r cos theta. So if you take a 90-degree here, this is your hypotenuse and this is your base, right? So if you want to find this P value, right? So P value will be your actually base by hypotenuse, right? So from P by R magnitude. Now that from there you can actually get this P equals to lambda dot r cos theta. Okay, so that is P equals to lambda dot r. Now this p direction, these are all vectors. So this is in this direction, q is coming in a negative direction. So the direction of q and p is different. For this q have a negative sign. So q is minus lambda dot r dot s. Now this r dot s is your dot product of this scattering vector, and this s naught is the initial, this is your final. So this you can actually find out with simple trigonometric measurement. P and Q value are this. So if you add on, P plus Q is equals to lambda dot R dot S naught minus S. Okay, so this is your path length. Now we are, if this is an electromagnetic wave, it will have an electric field and an magnetic field perpendicular to one another. Since we are here considering mostly the electric field generated by this one, so that energy, that electrochemical energy or that electric field generated at time 0, and this z is the path length. Okay, that can be given by this equation, that is a cos 2 pi by z, 2 pi z by lambda. Lambda is the wavelength of the light. Now this z is similar to your path length. This z is this path length. You put this value here. Okay, so this will be your 2 pi, means lambda r s naught minus s. So this lambda will cancel out with one another. This cos 2 pi will be actually present, and you will be coming to this equation where you are going into this 2 pi r dot s naught by s. Lambda will cancel out one another, and that will be 2 pi r dot s. Right, so this r dot s, this is a vector, right? And what is this s? This s can be, this is the result of the initial and the final scattering vector. Means if you draw this in a plane, this s vector is a s plus s naught, means s vector plus s naught vector gives you the final scattering by the electron. So the final scattering by the electron is determined by this electron scattering factor, that is this s. Now this s, if you take the magnitude, it gives 2 sine theta by lambda. So this is being determined. Okay, so what is the relation of this 2 sine theta by lambda? So you see this will actually lead to our Bragg's law. So we will be coming in the next slides. Okay, because S is equals to S naught divided by 1 lambda. So the capital S value is 2 sine theta 1 lambda. Okay, and if this magnitude of S or the order of this diffraction becomes 1, then it is become 2 sine theta, and it is going lambda, n lambda equals to what we have, this d is equals to n lambda equals to 2d sine theta, right? So that concept we will be coming from here. That is for the atoms. We now here we are for the electrons. Okay, so electrons we little bit understand, this is the scattering vector of the from the two electrons. The scattering that is occurring, that can be defined by this capital S, that is we subtract the S final S scattering vector from the initial S scattering vector, you will find the scattering vector of the two system electrons. Okay, now we are going higher. From the electrons we are going into atom. Okay, now in atom you have a lot of electrons present. Okay, so here we need to make a consideration that in this atom the electrons are centrosymmetrically related. Meaning that there is a center of inversion. If someone of you have looked into my last video, that where we have talked about this symmetry operators, there is this inversion, right? So the inversion center means if this coordinate here is x y z, sorry, if the coordinate here is x y z, okay, so sorry, here it will be minus x, minus y, and minus z. Okay, this means if these two points are connected, then this will form a inverted center, the center of inversion. Here we are considering the center of inversion as the nucleus of the atom, and the two electrons inside, means each of the electrons inside this atom are connected by, they are centrosymmetrically connected with one another. Okay, so now what we know is that if the two atoms are centrosymmetrically connected, then the electron density of that two atoms will be similar. The electron density or amplitude of two atoms which are connected by centrosymmetrical or inversion by an inversion center, the intensity will be similar for both of them. Okay, so this can actually help us to reduce the number of electrons, because we are considering or assuming, this is not a real scenario, but we are assuming that in a atom the electrons are centrosymmetrically related to one another. Now you have the electronic scattering, the energy related with this electronic scattering, that is this 2 pi r dot s, right? So this I r dot s, so if you solve it, you will get the relation of the phase. So here this is your actually phase, this will give you the information about phase, because r dot s will be r s cos theta. So anything angle where it is coming, it is actually the phase. Right. So then someone can ask that where from this exponential term is coming. So this exponential term is coming because if you put this 2 pi r dot s into this equation, where you have this cos 2 pi r dot s, right? So this cos 2 pi r dot s will be what? R dot 2 pi r dot s means r magnitude, s magnitude, cos theta. So cos theta means cos of p plus q, right? So this cos of a plus b means, if you solve this cos of a plus b equation, you can get 2 cos theta sin theta, right? Means what I am telling you, that from here you can go use an Euler's formula, where you can write this e to the power I pi, means I theta, e to the power I theta is equals to cos theta plus I sin theta equals to e to the power I theta. So from there only you can actually, you are getting this e to the power I is the this, and this angle, that is 2 pi r s, 2 pi r dot s is your phase angle. You are getting that e to the power I theta, that theta value is 2 pi r dot s. Now for each electron, the electron density is rho r, and this is your phase information, and nucleus is your center. Just like in the first, in the electron, there this electron was center, and you took positions of other electrons with respect to this electron. Here you were taking the nucleus as center, and now you are actually calculating the scattering by each electron, multiplying it with the intensity of each electron, means this is a unitary method, means this much intensity is for only one set of electron. Now you need to take the r, means the radius, to all those electrons which are present. So that's why you are integrating it, multiplying the electron density of one electron with the phase. Okay, 2 pi r dot s. Okay, you are getting into a something known as atomic scattering factor. So the small f is the atomic scattering factor, and these two electrons are actually connected by one another. You can represent this diagram in the Cartesian form like this. Okay, and here also this s value is the magnitude is written as 2 sin theta by lambda. So this is being experimentally determined. Okay, 2 sin theta by lambda. Now we are moving from the atom to a unit cell. Okay, so you don't need to remember these equations. I am just telling you to know from where these softwares have been evolved. Okay, if you don't remember, if you don't imagine it correctly, no problem, because finally what the logic should be clear. Don't go too much around into the mathematical equation. Okay, so if you are clear with your logic, that will be fine. Now So only two slides, or we can. So it's already 8. So what we can do is that from here, from the scattering of the unit cells, uh, okay, let me finish since I am actually talking about next slide. To next slide, we will finish. Then we will continue the next class from this concept of reciprocal lattice eval sphere. This we will be doing in the next class. So, so now we are from the atom, we are going into the unit cell. We are coming larger, right? So from the atomic scattering factor we got, okay. Now there will be different atoms inside this unit cell, and we take a origin present in this unit cell. Now in the previous one, the origin was the nucleus of the atom. Now, when you are calculating, you have a shift in the origin because of you are there are now individual atoms. So what we are doing here is that we need to take the individual scattering atomic scattering factor of what we found here, this F value of each atom, and we are relating one atom with one another through origin with the help of this phase 2 pi I rj. Rj is this distances of these atoms from the center. So this is your unit cell factor. Now when you actually take means this is for a single system of atom, right? Now if you take all the unit cells together, okay, and add them together, then you will get into your structure factor. So if you add all the atoms, all the means the total scattering from the unit cell, if you take and sum it, you will get your structure factor. So your structure factor that we were talking about is this Fs. So this is your structure factor. So this you actually get in the reciprocal plane, because this structure factor you will see it's a unit of HKL. So how that is so? And this alpha is your phase. Okay. And this F1, F2, F3 is the individual atomic scattering factor. So the idea here is that electron has an electronic scattering factor. If you add electronic scattering factor with the electron density and multiply it with the electron densities, individual electron densities, you get atomic scattering factor. Now, if you take all the atomic scattering factors, add them together, you get to the structure factor, which is the total scattering by a single unit cell. So this is your structure factor. Okay. Now we are in a unit cell. Now from the unit cell, we need to go and see how the scattering occurs in the three-dimensional crystal. So for that, now your unit cell will be related to another unit cell in the crystal, and there will be an origin which will connect, means there will be a frame of reference which will connect this unit cell with this unit cell, for which you need some vectors like this t dot a, u dot b, v dot c. So these are some values. Whenever you want to denote a vector in a three-dimensional space, you actually denote like this by a equation like 3y plus 3x plus 4y plus 5z. So this will give you a vector in a three-dimensional space. So this is a vector which is connecting this unit cell with this unit cell. So similar to, so here what we are doing is that the idea is there are n number of unit cells which will repeat itself in a x direction. There will be n number of unit cells. Since unit cells have to repeat itself, then only it will create an order. In the y direction, there will be n number of unit cells. In the z direction, you have n number of in unit cells. Okay, and all these unit cells, this n number of unit cells in x, y, and z direction, if you take all the individual structure factor of all these unit cells, you will actually get into the structure factor or the main scattering factor of the crystal. Okay, but now someone can ask me that, sir, you are telling that there are n number of unit cells in the x direction, n number y direction, n number z direction. What will happen if some unit cell is present here, not in the three-dimensional space? So for that you have this origin. So you need to connect this origin with this unit cell with certain vector. Okay, with the help of this coordinate you can actually get the structure factor of that, means that point in space as well. So for that we are actually doing this. So this is for x direction, this is for y direction, this is for z direction. Now there are n number of molecules present, n number of unit cells, n1, n2, n3. So we are multiplying all with this, and also this T, U and V will serve the this kind of relation between the means unit cell which are not in the x, y, z direction but in between. And then you are coming into this final Ks value. Now, if you see this A dot S, B dot S, C dot S, this exponential, this Ks is actually related to this exponential term. If this exponential term is very larger, okay, so then this Ks will be 0. Means if this exponential term reaches to infinity, then this Ks will be 0. Right. So if this exponential, so what, you don't require this ks to be 0, because the overall scattering, if it is 0, then you will not get any point. For that, to actually not getting this ks to be 0 and to get it into some value, this a dot s, b dot s and c dot s, this was found to be that if they are adding up to some integer, means if they are some integer, a dot dash is an integer, b dot s is an integer, c dot s is an integer, then this exponential term will not go up to that much higher value, due to which the ks will not be 0. From here actually the idea of your Law's equation came, that this a, that is the basis vector, dot s, that is the scattering structure scattering vector, is equal to h, b dot s is equal to k, c dot s is equal to this l. So these are your Miller indices. If this condition satisfies, that is your Lewis in condition, then only your crystal will be actually diffracting and you will get the diffraction spots in a constructive interference. Now from here, if you see that if 1 by s, this is the 1 by s, the value magnitude of the s is one, this denoted by this, and 1 by s was found to be similar to the interplanar distance. So interplanar distance is d. Now we know that magnitude of s can be written as 2 sine theta by lambda. Now you substitute that 1 by s, you do 1 by s here, 1 means reverse this equation, you solve it, you find, and if your this order of diffraction is 1, you found that 2d sine theta by lambda is equals to 1. And if it is ordered is of n, means n number of planes are is there, then it will be n lambda, that is 2 sine theta equals to n lambda. So from here you are actually getting this Bragg's law. So Bragg's law and Lewis condition point out to the same equation, okay, but they have different forms. Okay, so that this to this part we have actually talked. Now we will have to go to concept of reciprocal lattice, that is actually made up by this HKL values. So since it is already 8:6, we are not going to that reciprocal lattice and everything. So one thing what I wanted to show you is that since we have talked about the structure factor, so this is the structure factor equation that we got. Now the small dv can be replaced by the volume dx, dy and dz. These x, y, z are still in the means the real space, right? But this, when you are actually multiply it with some r dot s, means what is this r dot s? This is a dot x plus by plus cz. But this s value, this is not present in your real space. This is the scattering factor, and scattering factor is in the reciprocal space. So a dot s, b dot s, c dot s, we know from Lewis condition this should be integer, which are your Miller indices. So r dot s is hx plus ky plus lz. If you put these values in this equation, this is the structure factor relation, which is a function of your HKL, which is making in the reciprocal space by making a reciprocal lattice by this HKL. But we don't require the structure factor. We require electron density from this. So if we reverse this equation, or actually perform a reverse Fourier transform, you will find that the electron density is 1 by V HKL of structure factor exponential to the power minus 2hi hx ky lz. Now if you solve this, if fhkl is a vector, if you actually take the magnitude and solve this equation, you will find that again e to the power I theta, Euler's formula, cos theta plus I sin theta equals to e to the power I theta. So this theta is your phase, so this e to the power I theta, so this is your phase and this is your amplitude. This value you are actually getting up from this structure factor, and this structure factor will be directly proportional to the intensity. So if this structure factor, if you take the structure factor square, you will get the intensity of the spots. But to solve the final electron density, to get the final electron density pattern, we require an amplitude term plus a phase term. Amplitude term can be easily found by getting the structure factor, squaring it, you will get the intensity. But what is the phase? The information of this phase here we can't determine from this equation. We don't get this information. So how to determine this phase? So this was a real problem which was known as famous phase problem of structural biology. For this we have, means people have developed various methods that we will be actually, this is the importance of phase. So we will stop here by this example. So I want to show you how much phase is important. So this is a good example where you actually take the amplitude. So this is the dark amplitude and this is the means phase of the dark. This is of the cat and this is of the cat's phase. Okay. Now if you take the phases of the duck and take the intensity of the cat, it will form similar, a pattern similar to the duck. If you take the phases of the cat and intensities of the duck, something will look like, the final result will look like a cat. This is the importance of phase. Means the whatever may be the intensity, if you can't find the phase, you can't find the final structure. So for finding the phase, people develop different techniques like this, which we will be discussing from there. Here we will be starting our next slides. So direct method, experimental phasing and molecular replacement we will be talking in the next class. Okay. And also this eval sphere and concept of reciprocal lattice is left. We will be discussing this in the next class. Okay. I know this part is sometimes not very clear, but just I wanted to show you how actually this happens. So yes, I think you got some idea, right? Uh, okay. So you can go through these videos, lectures, and come back in the next week. We can discuss if you have any doubts. Okay. So I will be ending the class now. Do you have any questions, Shani? Okay, okay, fine. Then go through it and you can ask me in the next class. Okay, bye. Bye.